Hi, today we will see about apoptosis. Apoptosis is the process of programmed cell death. It is the term which is first introduced by Kir et al. In 1972. Apo means from and ptosis means fall. In Greek, the apoptosis is the fall of leaves or the petals. The apoptosis is the physiological process that is triggered by the activation of genetic self-destruction programs existing in the genome of all cells. It is the physiological process in which the cell death is brought about through a regular sequence of events. It allows for the programmed removal of specific cells without harming the nearby cells. In this way, the multicellular organism destroys the undesirable cells from your tissues. Any defect in this process will lead to the important role in variety of diseases. The apoptosis is an active form of cell death which is characterized by the biochemical and morphological processes, especially by the chromatin condensation, polynucleosomal DNA fragmentation, and fragmentation of the cells into apoptotic bodies. The apoptotic process plays a central role in the development and functioning of the immune systems too. Here you can see the apoptotic reaction in a normal cell will leads to the chromatin condensation. After the chromatin condensation, the membrane, that is the membrane, became blubbing. The blubbed membranes first appears, then it becomes a bubble-like structures, that is the apoptotic bodies. These apoptotic small fragments will be engulfed by the phagocytes. That reaction is called phagocytosis. The apoptosis can represent a defense mechanism at a cellular level against cancer by the participation of the proto-oncogenes and the tumor suppressor genes in the regulation of apoptosis. Each cell receives the multiple signals which by means of specific receptors can induce the cells to enter the cell cycle or apoptosis. The alteration of a specific receptors can lead to the appearance of the malignant clones due to the imbalanced relationship between the apoptosis inducing or the repressive signals and proliferation. The anti-apoptotic BCL2 protein may inhibit the apoptosis induced by the absence of growth factors, neurotrophic factors, and cytokines. Here the BCL2 is the B cell lymphoma 2, which regulate the cell death by either inhibiting, that is anti-apoptotic, or inducing like a pro-apoptotic. And here you can see the apoptosis reaction. It is the programmed cell death. When faulty enzymes or some other reactions will occur, means the cells undergoes some condensation, that is the chromosomal condensation, that is nucleus condensing. Some lobes, that is the blebs, occur. The blebs will become the small apoptotic bodies. These apoptotic bodies, that is small fragments, are engulfed by the scavenger cells, that is phagocytes. Then the apoptosis pathway. Apoptosis can be initiated by one of the two separate pathways, that is the intrinsic pathway and extrinsic pathway. Both these pathways ends with a final common effector pathway known as the execution phase. The intrinsic pathway The intrinsic pathway mainly triggers the apoptosis in response to the internal stimuli, that is, biochemical stress, DNA damage. This activates the P53 gene, which halts the cell cycle and initiates DNA repair. If this repair attempt is unsuccessful, means the apoptosis can be induced. Then the lack of growth factors. These factors are the internal stimuli for the intrinsic pathway. The intrinsic pathway is modulated by two groups of molecules like BCL2 and BAX. BCL2 is the B cell lymphoma 2, which regulates the cell death by either inhibiting or inducing apoptosis. BAX is the BCL2 associated X protein, which is the member of a BCL2 gene family. It acts as an anti or pro-apoptotic regulators. The activation of BAX leads to the formation of BAX-BAX dimer, which in turn enhances the action of a variety of apoptotic stimuli, which increase the cell susceptibility to apoptosis. That is, the cells are likely to be influenced for apoptosis. The BCL2 family consists of both pro and anti-apoptotic members, and it is the balance between these that determine how susceptible a cell may be to the apoptosis reaction. The balance between these groups of molecules establishes a molecular switch, which determines whether a cell will survive or undergo apoptosis in response to the internal stimuli. One is the extrinsic pathway. The extrinsic pathway triggers apoptosis in response to the external stimuli, namely the ligand binding at death receptors on the cell surface. These receptors are typically the members of the tumor necrosis factor receptor, that is TNFR gene family, such as TNFR1 or FAS. Binding at these receptors leads to the receptor molecules grouping up on the cell surface. It initiates the downstream caspase activation. Then the execution phase. The initiation of the apoptosis by either pathways results in cascade activation of the caspases, that is the successive activation of the caspases. The initiation of apoptosis first activates the initiator caspases such as the caspase 8, the role of which is to cleave other procaspases into active executionary caspases. These executionary caspases then cause degradation of variety of cellular structures such as the cytoskeletons, nucleus and other cellular materials. For example, the caspase 3 activates the DNase, which leads to the fragmentation of the DNA. This figure will explain you clearly about the all the pathways of the apoptosis. In the extrinsic pathway, the death receptor received by the cellular surface will, and along with these death-inducing signaling complexes like procaspase, and death receptors, etc. They will activate the caspase 8. The caspase 8 will activate the executionary caspases, and then they will induce the cell fragmentation and leads to the apoptosis. This process leads to a variety of morphological changes within the cells such as nuclear shrinkage, spignosis and fragmentation, that is karyohexis. Alongside this, cells itself shrink but importantly retain an intact plasma membrane, The dead cells are either immediately phagocytized by the neighboring cells or break down into smaller membrane-bound vesicles known as apoptotic bodies, which are eventually phagocytized. Then the role of apoptosis. The apoptosis play a major role in the embryonic development. The removal of cells where apoptosis is responsible for developing tissue forms and shapes, which includes various roles like interdigital cell death, which allowing the separation of the fingers during the embryonic development, and the cell death in the urachus, which allowing the removal of the redundant tissues between the bladders and the umbilicus. The urachus is the fibrous remnant of the allantois, and the cell death leads to the removal of the vestigial remnant from the earlier in the development, such as pronephros. The pronephros, the most basics of the three excretory organs that develop in the vertebrates. The first stage of the kidney development is called the pronephros. And the removal of the Mullerian or Wolffian duct when the sex of the fetus is determined. The Mullerian duct is the primordial cells of the female reproductive tract, and the Wolffian duct is the embryonic structure that form the male internal genitalia. And the cell death in the dorsal part of the neural tube during the closure. And the neural tubes, the embryonic precursors of the central nervous system. The removal of redundant epithelium following the fusion of palatine processes in the development of roof of the mouth is also a type of cell death. If the cell death, that is the apoptosis, fails in these sites, means like in the finger separation, like that everything means, or in other sites means, the variety of diseased condition will occur like syndactyly. It is a webbed finger condition. If the interdigital membranes or the interdigital tissues are not undergone the cell death, means the finger will be closed like a webbed finger, and a cleft palate or a spina bifida. The spina bifida is the in developing babies the spinal cord fail to develop or close properly. The apoptosis also play a role in preventing cancer also. If apoptosis is for some reasons it is prevented, means it can lead to the uncontrolled cell division, and it will subsequently leads to the development of the tumor. Thank you.